All right, can you see my
There we
screen?
go. Yes. All right. Thanks everyone for joining us for tonight's lecture. As a friendly as a friendly reminder, this lecture is being recorded. If you do not wish to appear on the recording, please keep your camera off. And to reduce feedback, everyone is muted upon entrance. If you have questions during the presentation, please utilize the chat feature and I will read them out when appropriate. But please know there will be time for Q&A at the end of the lecture. And with that, I will pass it along to you, Devin.
Cool, thank you so much for the intro. I'm super excited to talk to everybody tonight about my research. It's something that I care a lot about, super passionate about. And so today I'll be talking to you all about high resolution stream networks, revealing habitats for petition burrowing crayfishes. And this is part of my master's thesis at NC State. I've been studying these crayfish for about 2 years now, and I expect to finish up this year around December sometime. So I'm on the last leg and getting there. So first we'll start off with some background. There are around 669 species of crayfish globally currently described right now, more and more being described every year. And the United States harbors nearly 400 of those species, so a super great amount of diversity just in the United States alone. And the Southeast United States is a biodiversity hotspot for crayfishes as well, and there's around 332 crayfish in just these states alone. And the same can be said about fishes and mussels and other freshwater taxa as well. Okay, so we have all of this diversity in the Southeast, but what do crayfish do? What do they add to their ecosystem? Well, first, they are ecosystem engineers, and some crayfish can burrow into the ground. We'll talk about this a little bit later. But this burrowing behavior aerates the soils and creates refuge for other organisms, namely the crawfish frog. As you can tell with the name, they only use crawfish burrows as their habitat. And there's a lot of other species too that use crawfish burrows as refuge. And they're also trophic regulators. So they have this really unique function. They can function as predators, eating. They're very opportunistic, and most of them are very generalists, eating anything they can really get their claws on. And they can also function as prey as well, to a lot of aquatic species and also a lot of terrestrial species like raccoons, possums. They will eat crayfish as well. And then they also can function as shredders when they eat algae and leaf litter and then they poop it out. Smaller invertebrates will eat this as well, use this as food. So they have this really unique function in their system where they can be all of these things at once. And lastly, they can act as bioindicators. And this just basically means that the concentration of the nutrients in their tissues can be representative of the larger system that they're found in. So, for example, if they were caught downstream of maybe an industrial plant, we could take that crayfish, extract some tissue, and test that tissue to see what nutrients are in that tissue and the concentration of the nutrients in their tissue are can be representative of that system, of that stream that they were found in as well. So we can really know what's going on in their system without even testing the system directly. So despite this high diversity that we have in the United States, but also the Southeast, and their high ecological value, overall, as a taxa, they are very imperiled. So around 50% of North American crayfishes are considered imperiled to some degree by different conservation groups. And they're ranked third most imperiled taxa, right under freshwater snails and mussels. And despite this, there's a vast disparity in species recognized as imperiled by professionals, as we see here, and those actually listed on the Endangered Species Act. So overall, there's only 10 crayfish species that are federally listed on the Endangered Species Act, and we can see those listed over here. Overall, though, today we will be focusing on Mississippi crayfishes, and Mississippi includes 65 species and 5 subspecies of crayfish. 13 of those are endemic, which means they only occur in Mississippi and nowhere else in the world. And 17 of those are petitioned for listing under the Endangered Species Act. So we have a lot of potentially imperiled species in this state. And so we'll be honing in on 7 species of Mississippi crayfishes, and these are a species complex known as the rivulet crayfishes in the genus Hobsius. Like I said, there's 7 species in this genus, and all of them are endemic to Mississippi, and one of the species range extends into Alabama. Overall, though, they're all super cryptic, so they all look alike, and we can see some pictures here of what they look like. And they're very small bodied. In the picture to the right, we can see this is an adult male, and this is actually the largest one we caught during our sampling season, and he's not much bigger than a quarter. So super small, very cryptic species. And today I'll be focusing on one of the species of rivulet crayfish, the Octibaha rivulet crayfish, or H. orchnectoides. I'll be referring to it from now on, its scientific name. And you might be thinking, okay, what the heck is Octibaha? What does that even mean? It's actually named after the county in Mississippi that it was described in. So Octibaha County, Mississippi is what it's named after. And this is one of the Mississippi endemic rivulet crayfish. And we only have 14 localities for the species, ranging from 1915 to when it was first caught until 2012. And so we can see it in the map over here. Our pink triangles are the recent occurrence sites that we found the species at from 2007 to 2012. And then the green circles are those historic occurrences from way before 2007. And this is all we have. We have these 14 localities, and that's all we know them to be. And these are from 2 Huck 12 watersheds within the Tibby-Catalpa Creek watershed. And you can also see that depicted on the map here, how all of those sites are within those 2 Huck 12s, within those like black boundaries. And the focal systems that we'll be honing in on today we'll be describing some in a little bit, but all 7 species descriptions have some common keywords explaining their habitats. And some of them name specific habitats, but some keywords that they use are shallow, small, emergent vegetation, leaf litter, slow flow, and temporary. Keep the temporary in mind. But the specific habitats that they name are roadside ditches. We can see in a picture here, we have the road on the right side, and then our aquatic system, that little roadside ditch. They also name headwater streams as a habitat for these species. And then they also name larger creeks as habitats. So thinking back to that temporary keyword, they're referring to non-perennial systems or systems that don't stay wetted throughout the entire year. So we can see in this picture during the wet season in Mississippi is from January to May. This is a stream channel we caught rivulet crayfishes in. You can see that it's full of water during the wet season, of course. And then that same stream channel during the dry season, so in Mississippi from around June to December, it's completely dry. But these rivulet crayfishes are able to persist in these habitats. But how can they do that? Well, they can do that because, as we said before, they are burrowing crayfishes, and they're actually classified as secondary burrowers. And this just basically means they construct really simple burrows within the stream channel. So we can see in C here, picture C, that this burrow is going right into the ground, but it's located right within the stream channel, usually up against rocks or roots or things like that where they can have some cover. And they spend most of their lives in this burrow, but they often inhabit open water during the rainy season, which in Mississippi is around 9 months. And so that's why they're able to persist in these habitats that dry up temporarily. And they come out in the open water too to forage and breed and things like that. So, but how are they able to do this, right? They're living in the burrows. How are they able to survive? Well, they have external gills. As we can see in this picture here, those like fluffy pieces um, up against those like circular pieces there are the gills, and that's in the carapace of the crayfish, so like that body plate. And these external gills allow them to retain the moisture from the humid air, moist soil that they're in their burrow. And if they dig deep enough in their burrow, they can reach that groundwater level. And so they may be dependent on these factors for habitat suitability, which keep that in mind. We'll talk about that a little bit later. But super cool how they're adapted to do that. And so overall, we talked about the diversity of crayfishes, their imperilment, and the ecology of rivulet crayfishes overall, but why are we focused on rivulet crayfishes? Well, we have some conservation concerns here. Overall, the temporary nature of the non-perennial streams that rivulet crayfishes inhabit often makes them resemble dry land rather than flowing waterways. So we can see in this picture, this is actually a stream channel during the dry season, and you wouldn't even know by looking at this picture that it has water during the rainy season, and you wouldn't even know that it harbors aquatic species, right? So that's just one thing to look out for. And so, because of this, as a result of this changing appearance that they have, these streams are often excluded from maps, conservation assessments, they're undersampled, and they lack legal protection, thus leaving these really small but dynamic streams that hold a lot of diversity threatened by urban expansion and habitat loss. And because of the subterranean lifestyle of these rivulet crayfishes, they're hard to study when they aren't in the open water, right? And if they never emerge from their burrows. And so we're really worried about the data that we have on these crayfishes. And overall, burrowing crayfishes are super data deficient. We have that limited sampling period, and there's only one federally listed under the Endangered Species Act. And only 39% of them have life history data, but it's often incomplete. On top of rivulet crayfishes being burrowing crayfishes and inherently imperiled in that way, or inherently data deficient in that way, they themselves are also data deficient. There's only one article on one of the species in the genus, and we only have those few localities of H. orchnectoides specifically, but of all of the species in the genus as well. So really limited information. But why are we doing this research, which we'll be talking about in a second? In 2010, the Southeast Aquatic Species Petition was published by the Center for Biological Diversity. And this was a petition to list 404 aquatic, riparian, and wetland species under the Endangered Species Act. And included in this petition was my focal species, the Oktibbeha rivulet crayfish. So because they're petitioned for listing under the Endangered Species Act, we need to inform a species status assessment, which basically clarifies their range, their distribution, their threats, population, things like that. And we need information on their ecological requirements and all of these other things to make effective conservation decisions and to inform their listing decision upcoming in 2026. So the goal of my project was to really just expand the knowledge of H. orchnectoides distribution and threats. And we wanted to evaluate occupancy of rivulet crayfishes in various overlooked habitat types that we talked about earlier that were described in their species descriptions. And this is really where I come in. So we'll talk about my methods. The study range was within the potential range of H. orchnectoides. We saw this picture a little bit earlier with the recent and historic occurrence sites. And on top of those two HUC-12s that we know them from historically, we included the other HUC-12s within the larger HUC-10 watershed. And this really encompasses the potential habitat area that they could be in. So those 5 HUC-12 watersheds in 3 counties in Northeast Mississippi is our entire range. And so now we'll get kind of a boots on the ground look of our habitat types. And first we have our roadside ditches. We saw that picture of them earlier. They're running right up against the road, of course. And because of this, they're often heavily managed. So we can see here there's not really a lot of riparian area. There's not any tree cover at all. They're usually mowed, and this is why. And because they're often used as flood control as well, they're dredged out. And so we can see there's not microhabitat in the stream channel itself either. So just not a lot going on really in this habitat. And this system is non-perennial. And then we have our unmapped streams, which similarly are non-perennial in nature. And these are called unmapped because they are not depicted in the USGS National Hydrography Dataset. And we'll take a look at that a little bit later. But these are going to be our smaller, unnamed, undepicted tributaries that come off the larger stem systems. And we can see though, even though they're non-perennials, like our roadside ditches are, they have a lot more riparian area. There's herbaceous ground cover, and you can't really see in this picture, but in the stream channel itself, there's a lot of microhabitat. So there's algae, there's herbaceous vegetation, different substrates, things like that. And we have a lot of canopy cover, which allows for a lot of leaf litter input into that system for food and cover as well. And then lastly, we have our map streams, which are called mapped because they are depicted on the USGS National Hydrography Dataset. And so therefore, these are going to be our larger systems, our relatively perennial systems. And you can see it in this picture that has deeper surface water. And we can see though there's still some tree cover going on, there's algae, so enough food and cover coming into that system as well, but overall a better suited system for larger predators such as fish to persist in that habitat. And so to identify these sites for our roadside ditches, we georeference both sides of the road to depict potential roadside ditch habitat. You can see that in the picture here in the purple. That's what we use to depict those ditches. And then to identify our mapped and our unmapped stream sites. So first we'll start with our mapped. And like I said before, these are depicted in the USGS National Hydrography Dataset. So we can see those in the blue here are map streams, those larger main stem streams. And then we use GIS and LiDAR data to develop a high resolution stream network that is able to depict these smaller unmapped streams. And so we can see those depicted here in the green. So just from a side-by-side of these pictures, we can already tell that our unmapped streams are adding a lot of potential stream length to the landscape. And this is just one portion within my 5 HUD-12s. So we can already kind of see how much potential stream length that's adding. But we really just wanted to know if rivulet crayfishes are utilizing these unmapped streams and the difference in occupancy between our map streams, our unmapped streams, and our roadside ditches. So in order to do that, of course, we had to sample for crayfishes. So we did this from February to May of 23, the wet season or the rainy season in Mississippi, when our rivulet crayfishes were out and breeding and foraging in the open water. And we used a 2-pass removal method with 2 dip netters for 10 minutes. And we did a seine pass for fish. So this basically means we went out, 2 dip netters went out into the stream or the ditch. We did a pass of 10 minutes, collected anything we could. And if we didn't detect rivulet crayfishes on that first 10-minute pass, we did an additional 10 minutes. And if we didn't detect them on that additional 10 minutes, we assume they were absent, and we'll make an occupancy model in the future, but we'll get to that in a little bit. And then also, one of my thesis questions is focusing on predaceous fishes and if they have an impact on occupancy of rivulet crayfishes. And so for that, we seined for fish then as well after that. And if we were lucky enough to capture our rivulet crayfishes, we recorded capture counts, life stage, sex, carapace length, and male reproductive form. And we also collected genetic samples of at least 7 or a maximum of 7 rivulet crayfishes if we caught that many. And this included snipping their walking legs. And then we also collected 1 sexually reproductive male body for a morphometric study as well, if we were to catch one of them. So both of these to be used for genetic and morphometric studies. And then we preserved or noted all of the other non-target specimens that we caught for later ID in the lab and put those into a database. And then after we collected all of the information on our rivulet crayfishes, we returned them to the stream alive. And we also took some habitat assessments here. So we did a very extensive habitat assessment. We took water quality data, all of that information that you could think of. We recorded in-stream variables, so substrate, and in-stream cover, vegetation, pine straw, leaf litter, things like that. And we measured riparian area immediately outside of the stream or ditch, and canopy cover as well. We also did a SARP modified barrier assessment for my my 3rd chapter of my thesis, which is kind of looking into threats of barriers to aquatic passage. And we also installed monitoring wells in 50 roadside ditches in January of 23, so before our sampling season, to take a look at groundwater levels and inundation frequency of our ditches. We'll be looking at a little bit later too. So for results, what did we figure out? We sampled 185 sites from February to May of 23, and this included 64 ditches, 35 map streams, and 86 unmapped streams. And we caught more than 4,000 rivulet crayfishes. We can see them all piled up in these trays here, and one caught in a little dip net. Here's a map depicting the sites that we sampled. So the blue lines are the NHD map streams. The green lines are the high resolution stream network that we developed. And then within our sites themselves, our roadside ditches are the purple triangles, our map streams are the green circles, and then the unmapped streams are the red squares. So we can see pretty widespread coverage over our range overall. The only small little gaps that we have are because there was no road access to those areas. So the sites that we were sampling were directly either next to the road, of course, for roadside ditches, and the streams were intersected with a road. So in those little gaps on like the top right, just because this is such a rural area in Mississippi, there was simply just no road access for us to even like access those areas. But pretty widespread coverage, and we can see kind of the red is taking over. We ended up detecting rivulet crayfishes at 64 sites, which is around 35% naive occupancy. And this included 9 roadside ditches with around 14% naive occupancy, 17 map streams, around 49% naive occupancy, and 38 unmapped streams, which are around 44% naive occupancy. And here's a closer look at where we caught our rivulet crayfishes. And so these are all the occurrences, same depictions as we saw before. And now we can see definitely that the red squares, those unmapped streams, are really taking over, not only because they're red and we can see them easily, but also because they are just so abundant in this map. So we can talk about that a little bit. We asked, our question, right, was, does occupancy of rivulet crayfishes vary among these overlooked habitat types? And we found that it did. So pairwise t-test that we ran showed occupancy between unmapped and mapped streams differed significantly from ditches, but not from each other. And we saw that kind of in the naive occupancy rates as well. So the probability of presence of rivulet crayfishes at ditches is significantly lower than absence. So when we're pulling up to a roadside ditch, we're a lot less likely to find them in that system. And we can kind of see that depicted in this graph here. We have habitat type on our x-axis and probability of occupancy on our y-axis. And then those red dots within the dot system for each habitat is the predicted probability of occupancy. So we can see the predicted probability of occupancy in our ditches is a lot lower than the probability of our mapped and unmapped streams. And when we look at our mapped and unmapped streams probability, though, they're pretty comparable, almost the same, around 50%. And this may indicate that ditches may be less suitable habitat for rivulet crayfishes. So keep that in mind, though, and we'll just touch a little bit on abundance of our crayfishes in each habitat type. Even though we only detected rivulet crayfishes at 9 ditches overall, right, really low occupancy rate, we found in one roadside ditch, we caught 496 rivulet crayfishes just within that 10-minute sampling period. And the total overall that we caught for the sampling season was 795. And this was is the picture of the ditch that we caught those almost 500 in. And you can see them all in the little trees there, super small. Compared to our unmapped stream, which really comes up on top, we caught just within a 10-minute sampling period in one unmapped stream more than 1,000 rivulet crayfishes, and you can see that pictured here. And the total amount caught for the sampling season was nearly 3,000. And then compared to our mapped streams, in one mapped stream, we only caught 57. And the total for the sampling period, the whole sampling period, was only 310. So we have some conclusions here, even though, so our unmapped streams are valuable habitat for our rivulet crayfishes. The data is telling us that. And the unmapped streams are comparable in occupancy rates and are coming up on top in abundance, yet they receive less focus than our map streams do, which are depicted on our maps. They receive protection, so these systems that are really overlooked are valuable, just as valuable to our rivulet crayfishes as these map streams are. And rivulet crayfishes are more prevalent than historically reported. When we started, we only had those 14 localities from a very, very long time period. And we ended with 64 localities, right? So we're finding these species in a lot of areas. We just need to look at the right timing when these species are out in the open water. And these overlook systems add a lot of potential habitat area to the landscape. So just within my 5 Huck 12s, the roadside ditch system adds 554 kilometers. Our unmapped stream layer adds 1,400 kilometers. And then if we compare that to the map streams, they only have 363 kilometers of stream length. So these overlook systems are adding a lot of potential habitat area that we can see are valuable to our rivulet crayfishes. And we're still curious though, like what's going on with these ditches? Why are we not finding rivulet crayfishes in ditches, right? We only found them in 9. And so super low occupancy rate, but we're finding them in high abundances in the ditches that we are finding them in. So what is different about these 9 ditches that we caught them in versus the 50 something that we didn't catch them in? We're curious. And so if we remember back, our rivulet crayfishes need access to groundwater or moist soils in their burrows to avoid desiccation during the dry periods, right? So we hypothesized that maybe groundwater may influence occurrence of rivulet crayfishes, and that more frequent or longer durations of shallow groundwater levels may be more suitable for rivulet crayfish occurrence. Um, so we'll take a look at some prelim data that we have for that. And so the burrowing depth for rivulet crayfishes has been observed to be no more than a foot, so about 0.3 meters. And that was written in their species description. And I've also personally dug out rivulet crayfish burrows and observed the same pattern. So let's look at this graph here. We have number of days that the groundwater falls below 0.5 meter, which is that threshold that we set on the x-axis. And then the y is the occupancy probability of our rivulet crayfishes. And so we can see here it's really the exact opposite of what we hypothesized. We thought that they would like shallow groundwater for more days out of the year. And this graph is just during the dry period. So when these species need this groundwater the most, we hypothesized. But if it's looking like the larger the number of days, the higher these number of days that the groundwater falls below the threshold that they can reach, there's a higher occupancy probability. So the exact opposite of what we thought. And this is frequency. And if we look at duration of the longest period, it's the same type of trend here. So that's super interesting and not what we thought. So we might start to hypothesize what makes a roadside ditch suitable for rivulet crayfishes. That's to be determined. We have these results that groundwater might not be influencing their occupancy in these ditches. And so they might depend on other soil characteristics for their persistence. We know that somehow they are persisting in these dry systems when we can see now that they don't have access to groundwater. So how might they be doing that? Some other ideas are soil moisture content, soil texture, and soil compaction. And these are all things, all factors that either we took data on in the field or we can get them remotely as well. So all things that we can test to try to figure out and really pinpoint the difference between these 9 ditches that are holding rivulet crayfishes and the 50 odd something that aren't. So stay tuned for that. Just some preliminary stuff that we've been finding. And overall ideas and next steps in general. My next step is to make my occupancy model, taking into consideration detection rates and other habitat variables, and testing these habitat variables for association between occupancy of our rivulet crayfishes. And one thing I think that might influence occupancy is microhabitat in these systems. So as we saw in our picture of our unmapped streams, there was a lot of riparian area, a lot of canopy cover, and so a lot of ways that the cover and food can come into the system as well. So we can see in these pictures is a close-up of sites that we caught rivulet crayfishes in. We have a lot of herbaceous vegetation, like emergent vegetation in the stream itself. And we have a lot of leaf litter and root wads, root mats, things like that, where they can hide under and get away from predators that live in these systems. And we can see some algae too as well. So that just might be something that I think could influence occupancy. Stay tuned for that as well. We'll see. With that, I'd like to acknowledge my funders for my project, U.S. Fish and Wildlife, USGS, for having me as a student, NC State also for having me as a student, and all my project partners at Mississippi State, University of Mississippi, and my whole field crew, everyone who has helped me along the way. And with that, thank you so much for coming to my talk, and I will take any questions that anyone has.
All right. Thank you, Devin. That was super informative. I personally did not know there was that many different crayfish in the world, nor that you could catch that many crayfish in one go. You said 10 minutes you caught like 1,000 crayfish.
Yeah.
That's that's wild. Like we've
Yeah.
been, and these are, was that in one of the roadside ditches?
Hmm.
Yeah, like I have taken kids out into streams before and we catch like crayfish, and maybe it's because we have kids who are like scaring them away. I don't know. But that's truly impressive.
Yeah.
Um, Raylene is asking, how do you get the crayfish out of the
Oh, okay. That's a great question. So in my case, we sampled these crayfishes when they were out in the open water. So we actually used a dip net to sample for them, just a regular fish net. So we didn't have to actually dig them out of their burrows. However, for species that don't come out of their burrows at all, and some crayfish don't, they live all of their lives in burrows, we do actually have to excavate them out of their burrows. So oftentimes we take a couple things out into the field with us when we're digging for crayfishes that live in their burrows. We take a soil knife, which is basically just like a little shovel knife type thing with like a serrated edge that you can cut roots around. So if you're trying to like dig out a burrow and there's roots, you can cut them and dig out the soil with just like the knife part of it. We also have crayfish pumps, which are super cool. You can like redneck makeshift them, or they actually like sell them online. I'm not quite sure what they're called. They're definitely not called crayfish pumps. But if there's water in the burrow, you can actually like pump out the water and bring the crayfish to the top. But usually we use just shovels. When we see a crayfish burrow, just use a little spade shovel to dig around. And then we just like actually reach our arms down into the burrow and excavate it with our hand to grab that crayfish out. And yeah, and get that crayfish. But they're solitary. So there's usually only one crayfish in the burrow. And sometimes it can take an hour to a couple hours to dig some crayfishes out. So it really depends. But yeah, crayfish burrow digging is super fun and you get so muddy.
Do you have any recommendations of like what to look for are when you like look at a space and are like, I bet there's a lot of crayfish in that space? Like, what are the signs?
Yeah, like when you're looking at like a stream or what type of habitat?
Stream or a ditch or like what are you looking for any of our people watching who are like, I want to take kids to go find as many crayfish as possible? Like what are some like hallmarks that you can to find?
Yeah, that's a good question. So I will say one thing. I mean, crayfish are very robust and they're very widespread. So usually when you're walking up to any aquatic system and you're dipping around with a net, one of the taxa you will probably find is crayfish. And it just depends what kind of system you're in, whether you find a lot of different species of crayfish or just one species really dominates that system. Um, it depends, but usually some good indicators of good crayfish habitat are a lot of deciduous tree cover. So, like I said, they crayfish really eat anything, but they leaf litter is one of the things that they eat. And so, if there's a lot of leaf litter coming into that system for them to feed on or take cover on, they like that. Herbaceous vegetation too that they can eat or take cover in. And usually, when you go into a more shallow system that doesn't harbor a lot of those larger fish predators, you're probably better off finding them in there, or they won't be like hiding at least.
Yeah.
Yeah.
I don't see a last call for any questions here. What are you, I know you were talking about like your next steps. Um, what are you like, I mean, you probably said this earlier. Um,
It's okay.
Like, what are you doing with the crayfish data? Uh, like for counting how many crayfish are in
Yeah. So with all of the information that we get, ultimately it will inform the species status assessment for this species or for my focal species. So within my range that I sampled, there are potentially three species of rubullet crayfish within that range. And so, like I said earlier, we were taking genetics to kind of clarify the distribution and taxonomic revisions of the rubullet crayfishes in that range, and ultimately inform their species status assessment under their listing decision in 2026. So, all of this information on their habitat, their abundances in these systems, where they are, their range and distribution will go feed directly into informing that and further conservation decisions if need be.
Thank you for clarifying.
Sorry, repeat that.
I said thank you for clarifying that for me.
Yeah, of course. Thank you.
Question here.
Sorry, you're breaking up a little bit. Did you ask me something else?
No, I think we might have one more question here. I just
Okay.
saw the go. Well, if there aren't any other okay, so we're good. Well, if there aren't any other questions, we can go ahead and wrap this up. Thanks everyone for tonight. Thank you, Devin, for presenting. Oh, there's the question. What body parts do you take off the tissues to study?
Oh, so you can do it two ways. The way that I did it was snipping their walking legs, which they grow back actually. Um, so we took just like little clips and just took two of their walking legs and preserved it in 95% ethanol to extract genetic data. And you can also take gill tissue too. So you can go with forceps into their carapace, just grab a little bit of their gill tissue, take it out, and then preserve it in that same way. I don't know what the pros and cons for doing either of those are, but I know those other are the two ways that you can do it. Yeah, thank you.
Okay. Let me take that again. All right, thanks everyone for tonight. Thank you, Devin, for presenting. For those seeking EE credit, you will receive a follow-up email later in the week or early next week with the attached EE form. We will also be making this recording available on our Environmental Lecture Series YouTube playlist. We hope to see everyone again during our next lecture. We're going to have Katie Mangum from Bluestem Cemetery, and she's about natural burial and how it can integrate with land conservation efforts so that even after you die, you can still be making a positive impact on your local environment. So I hope you guys will join us for that. That will be June. Again, thank you, Devin. Thank you, everyone, and have a great night.